Hello, everyone. It's okay to start? Hello?
Hello, Dr. Belen.
Hello, Marco. So should I start? Okay.
Maybe you can start your lectures right now as we're waiting for other participants join the classrooms.
So should I start or not? Or waiting? I don't get it. You say should wait a little bit?
Yeah, maybe we should wait for a bit until maybe some participants join the meeting.
Okay. Okay, I think I will start because we are like a little bit late. So anyone joining, they can keep joining while I start introducing the lecture. Okay, good evening, everyone. Welcome to lecture number 6. But before we start, I want to say something. This is environmental monitoring in the real world lecture, but as today we are part of a special program, the University Global Classroom. So today we have participants from environmental pollution course and also from this special event. Welcome, everyone. Um, First, a little bit about me for those who don't know me. Let me introduce myself briefly. My name is Maria Belen Alfonso. I'm an associate professor from Kyushu University in Japan. My work and research work is mostly in plastic pollution, mostly microplastic pollution in lakes, oceans, and in food webs. I started working in Argentina I did my PhD and postdoc in lakes from Patagonia, and now I'm working at the Center for Ocean Plastic Studies from Japan, but doing some research project based in Thailand. I also work with other international initiatives, such as the Atlas of Ocean Microplastics, which is an initiative of the Ministry of Environment Japan. Also work with the Global Partnership on Plastic Pollution and Marine Litter from UNEP, and the Integrated Marine Debris Observing System, and also lead a network called MAPA, which is a national microplastic monitoring network in Argentina, among other projects. So during this class, you will see examples from my own field work in Argentina or in Thailand. And I want you to see how monitoring is actually done in real life, not just in the textbooks, okay? So what you'll be able to do after today's lecture, by the end of today, you will be able to do three things. First, tell the difference between monitoring and experiments and when to use which one. Second, to recognize the 5 key design decisions behind any monitoring study. And number 3, this is the most important one, you will be able to read any pollution paper critically. This means that you will know what questions to ask about how to the data that was collected. And this is the real skill. Every environmental decision in your country and in the world depends on monitoring data. So after today, you can judge that data. So this is today's agenda. We have until almost 9 p.m. I will try to do it more briefly. We start with the difference between monitoring and experiment. Then we will cover the 5 design decisions. Then cover about bias and uncertainty in research measurements. Then a real example about my project in Thailand. And finally, we will close with next activities. Also, we will have 3 small activities, 2 in the chat. You will be able to answer these activities. Mostly will be keep things very simple, just using the tools from the Zoom, like the chat or the reactions to answer the questions. But don't worry, every time I will explain every activity and what you will need to do. And if something is unclear, just you can type any question in the chat. I will be checking the chat on live. So we have a mixed audience today. We have students and also people from the university. So before we start the content, I want to say something about this is a special class, as I mentioned before, with a mix of purpose. On one side, we have the regular students for environmental pollution course, and also we have the people invited. So this is the lecture number 6 with me in this course, and they have already had 5 lectures. We are preparing for the last lecture also from next week. On the other side, well, we have the visitors from University Global Classroom Program. Welcome to all of you. For the visitors, don't worry, this is a lecture is very self-contained. You don't need to know previous content from the classes. So first, also I want to thank to Professor Dita and Professor Sapto for giving me the possibility to join these lectures and organizing the event. So let's start with monitoring experiments and why we measure. So what is monitoring? Monitoring is quite simple. You go out, you observe, you measure, and you do not change anything. Example, you will go to a river, you take a sample of water, you measure the plastic pollution, the microplastics, and then you come back the next month again and again. That is monitoring. You watch the river, but you don't change it. So why do we do this? This is because of 4 main reasons. To detect contamination early, to track any trends. Is the pollution getting better or worse on time? to check if regulations are working, and also to build any baseline data for future studies. Without monitoring, we have no idea what is happening in the environment. So now experiments. Experiments are different because in an experiment, you can change something on purpose to see what happens. For example, imagine you have 3 tanks with mussels. In one tank, you have clean water conditions. In another tank, you put some microplastics or plastic pollution. And in another tank, you put a lot of microplastics. And then you wait, for example, 4 weeks and measure how the mussels going, how they grow. So you change, in this case, the plastic level, and you measure the response of the mussels. That is an experiment. Why we do experiments? Because we want to test the cause and the effect directly, because we can isolate one variable or many variables to see the impact. And to test any scenario that do not exist in nature. For example, different concentrations of a pollutant or a chemical. So let's compare them side by side. So comparing monitoring and experiments, monitoring happens in the real environment. This is where. And in the case of experiments, this happen in lab control conditions, or maybe in the field, but under control conditions. Also, what you change. In the case of monitoring, you change nothing. But in the case of experiments, you can change one or multiple variables that you want to observe. When it comes to the strength, monitoring is strong in the real world relevance, but experiments are very strong in proving the cause and effect or something. And when it comes to the weakness, the monitoring is weak because at proving causation, but experiments are weak because they might not reflect the reality. Sometimes in experimental conditions, we use concentrations of a pollutant that we actually cannot find in the environment. But the idea is to see what happened in the future. An example of a monitoring is a beach plastic survey. And in a case of an experiment, a mussel exposure trial, like the one we commented before. Neither one is better, not the monitoring, not the experiment, but they answer different questions. So if you only want to know what is happening in the environment, you need to monitor. But if you want to know why it's happening, you need to do experiments. So this is a case example of my own research. This is a big one. In 2024, I launched this project in Argentina called MAPA. The full name is Monitoring and Analysis of Plastic Pollution in Aquatic Environments. MAPA was the first comprehensive freshwater microplastic monitoring network in South America and the whole continent, I would say. This is a rare case study. So we have more than 100 study sites across the country from the main watersheds in Argentina. We have more than 50 researchers from Argentina working in aquatic science from many different institutes and research centers across the country. And we study the microplastic pollution in lakes, in shallow lakes, in reservoirs, in rivers, and streams. And why does this project exist? Why so much effort? Because before MAPA, before this project, the freshwater systems of this entire country were almost unstudied, no one doing any research. So microplastics have been measured a lot. A lot of studies were done in Europe, in North America, in South America. No, nothing, almost nothing, or very few studies. So could we have answered this with one experiment? No. Could one science do it alone, explain the microplastic pollution across one country? No. But a coordinated monitoring network could fill this gap. So we use a standardized method. We work together to monitoring the concentration of microplastic in these freshwater environments. This is one of the first reasons why monitoring matters, because it will tell us what is out there at the scale of a single lake or at the scale of the full country. So in real research, monitoring and experiments work together. First, monitoring detects the problem. And then, for an example of a monitoring could be the high plastic pollution in Jakarta Bay. Then an experiment will test the mechanism. Does those plastic actually harm the organisms living in Jakarta Bay or the humans? Then monitoring will measure the response after an action. For example, did the plastics bag ban help? There was any change after this change? So it is a cycle. We first monitoring and explore, we then do some experiments or change something and then monitor again. In this course, we focus mostly on monitoring because monitoring is what most environmental decisions are based on and is what you will design for the next lecture. So now an important question, why does monitoring matter beyond science? Because monitoring will feed policy-making process. And policies are what is shaping the world. Let me show you two big examples. First, we have the GPML database from the United Nations Environmental Program. GPML has a digital platform with each country has a dashboard, and it collects the monitoring data from all over the world. Each country has their own information collected in this website. And this information is feeding, for example, the negotiations for the Global Plastic Treaty that is under discussion. Without monitoring data, there is no treaty. There is no regulations to abate the plastic pollution, to control the plastic pollution. A second example is AOMI. AOMI is the Atlas of Ocean Microplastics. This one is developed by the Japanese Ministry of Environment and Kyushu University. We, me and my colleagues from the Center for Ocean Plastic Studies, we support this activity. AOMI, for example, has a lot of measurements, microplastic measurements floating in the oceans from all over the world. And what they do is to standardize the data so it's comparable. So you can compare data from different studies from all over the world, and the policies and the government can use this information to make the policies. Without monitoring data, there are no targets. We cannot decide how much pollution, how much microplastic pollution or plastic pollution is too much. If we don't have this information, it's impossible to make any policy making. So now let me show you a study that became very famous in the news lately in Indonesia. Maybe you already heard about it very recently. In October, a researcher from BRIN, the National Research and Innovation Agency from Indonesia, published findings from 3 years of rainwater sampling in Jakarta. His name is the Dr. Mohamed Reza Córdoba. Maybe you heard about him. He has been working on plastic pollution in Indonesia for many years. He's quite famous. What did he find? He found microplastic in every single rainwater sample since 2022. They found 15 plastic particles per square meter per day falling in the coastal area of Jakarta. They found different plastic polymers from clothes, from car tires, from burned plastics like polyester, nylon, polyethylene, polypropylene, polybutadiene. And in January 2026, he had an update because after heavy rain, temporarily the air gets clean of microplastics. But the first rain after a dry period is the one who has the most concentration of microplastics. And why is this study important? Because it triggered a public health discussion across Indonesia. The health ministry advised wearing masks during rain events after this study. So a monitoring study became national health policy in only a few months. So what does the Jakarta study teach us? We have 3 main lessons here. One, the long-term sampling is very important. 3 years reveal the patterns that a single sample or a single measurement could never. Number two, the method of choice defines what we can see from the environment. In this case, for example, the researcher Cordova used special filters designed to capture the particles floating in the air or in the rain. But with other technique, maybe he cannot see nothing. And number three, monitoring connects the policy directly. When the data is communicated in a well manner, governments will act accordingly. This is exactly what monitoring is for and why it's so important. So now let's do the first activity. I will read four short scenarios. And for each one, I want you to type M if we are talking about monitoring, and E if we are talking about an experiment. I want you to put the letters in the chat so I can check, okay? So ready? Here we go. Okay. Scenario number one. Bring samples rainwater in Jakarta every week for 3 years. What do you think? This is a monitoring or is an experiment? You can start putting in the chat. Okay, very good. Marco says it's a monitoring. Anyone else? Abdul says monitoring. Muti says monitoring. Anyone else? We have like 34 people here. This chat should be full of letters. Okay, maybe let's try with the second one. A team adds plastic pellets to a fish tank and measures the growth of the fish. What is this? A monitoring or an experiment? Remember to put letter M for monitoring or letter E for experiment. You can make mistakes, don't worry. No one? Okay, let's continue. Number three, researchers expose mussels to 3 doses of microplastics in the lab. This one is easy. Do you think it's a monitoring or an experiment? I want to see the students from environmental pollution answering this. Come on. E, okay. E, good, very good. And number four, AOMI compiles microplastic data from 50 research vessels worldwide. This is monitoring or experiment? Marco is confused. Experiment? Maybe. Anyone else? Experiment. Anyone else? Well, this one is a monitoring case because they are compiling the data from different research vessels across the world. So they are doing a monitoring in the ocean. Remember that an experiment is when we can change a condition to see what's happened. But here, nothing is happening. I mean, nothing is changing. They just go there and measure. In that case, we talk about monitoring. But in the case of the plastic pellets in a fish tank and measure the growth of the fish. That is an example of an experiment. Okay, let's continue. So now we move to the core of monitoring. Every study has to make 5 main decisions, and we will look and discuss about each one. So the decision number 1 is the good question. Every monitoring study starts with one question. What do you want to know? For example, we have here 2 examples, okay? Look at the 2 examples in the screen. On the left, we have a sentence that says, I want to study plastics in semarang. This is really vague, really general. I want to study plastics. But this is not a question. This is a topic. You cannot design a study from a topic. This is too general. On the right, we have something more clear. How does the microplastics accumulation differ between Pantai Marina and Pantai Tirang in the rainy season? This is a real question. It tells you where to sample, when to sample, what to measure, and how to compare. If you cannot write your question in one sentence like this, your study designs will be very confusing. So decision number 2 is where. We talked a lot about these topics in previous lectures a little bit. Where you sample will change everything you find. And here we can talk about 3 subsections. One is the scale. Remember the scale matters, the lecture we had before? Do you study one site or multiple sites, or you measure in a full bay, for example? Another thing to consider is the location. Do you go to a hotspot of pollution, a place where there is a lot of pollution, or you measure also in a background site? A background site will be a place with no much pollution, or maybe both. And another thing to consider is coverage. How much of the area is represented by your samples? Are you going to measure all the area or maybe just a small portion? Think about Semarang, for example. A study at the mouth of the Banjir Canal will show you very different plastic loads or concentrations than a study if we go 5 kilometers offshore inside of the ocean. The concentration of plastic will be completely different. But both are in Semarang, both are talking about Semarang plastic pollution, but they will tell very different stories. So the second thing that we need to consider is where we measure. Then we need to talk about when to measure. Time is just as important as the place. Here we need to consider 3 things. One is the duration. Are you going to measure one day, one season, maybe multiple years? Another thing is the frequency. Are you going to measure daily, every week, every month? And the timing. For example, are you going to measure wet season or the dry season or both, or maybe day during night, or maybe you consider the tide, you can measure during the high tide or low tide in the beach. An example could be a beach survey in Bali after a single storm will show a lot of plastic, but the same beach in the dry season looks almost clean. But here we are talking same location, different time, and opposite conclusions. That's why when to measure is very important. Let me give you another example of why timing matters. This is one from my own research ongoing in Thailand. The results I will share are very preliminary because we are still analyzing these samples for a publication. But we are here, for example, we monitor the Bang Pakong River. Bang Pakong River is one of the main rivers flowing through the Gulf of Thailand. It's a very big river. We sample the microplastics floating in the surface of water during the dry season and the wet season during 1 year. In the photo, you can see the equipment we use. It's called a manta troll, a manta net. It floats on the surface and collects whatever passes through the thin mesh of the net, collecting all the microplastics and maybe some debris, maybe some plankton. Everything is concentrated. Before going to the field, we have a clear hypothesis. We expected to find more microplastic during the rainy season. But why? Because the rain will wash the plastic from the land into the river, also maybe from the atmospheres, as we saw in the Jakarta Bay study. So the heavy rain season means that they will have more runoff from the coast to the river. And this means that more plastic is able to reach the water. That was the logical prediction. It is what most scientists will expect from this monitoring. But when we look at the data we get, we found actually the opposite. We found that the concentration during the dry season was much higher than during the wet season. What this teaches us is exactly the lesson of today. First, what you find depends on when you sample. If you can see in the graph in the left, we have the dot line. That is the day I went to the river and measured the samples during the wet season and the dry season. And the other graph is showing the precipitations, the rain, the daily rain during that year. So a single dry season sampling will tell one story, but a single wet season sampling will say another story. But neither of both, none of both are true, I mean, by itself. So your hypothesis could be wrong, and this is not failure, this is science working. We measure to find out, not to confirm what we already think. So we were expecting the higher concentration in the wet season, but actually we find the opposite. To understand why this happens, we need to look at more factors, different factors. In this case, the one thing that you need to consider is that, as you can see, the heavy rain started in Thailand in May 2024, that year. And the most heavy rains, the heavy periods, already happened a few months ago before the survey. That means that maybe most of the microplastics already washed away from the land to the river. And when I went there in July to measure, the microplastic concentration was lower than expected. And in the case of the dry season, there was mostly no rain. So the concentration of microplastic in the water maybe was higher or similar to the wet season. And the pattern we were expecting didn't happen. And this is explained because this depends on the day we want to go measure. Maybe if we increase the number of measurements, one in May, one in June, one in July, one in August, we will find an increasing concentration during the wet season compared to the dry season. But sometimes because of budget, because of timing, or because the analysis of samples, it takes too much time. We sometimes have to make decisions and we have to decide maybe to do one survey instead of many. So the decision number 4 is how we measure. The method must be standardized. This means that we use always the same protocol, the same equipment every time we go to measure in the environment. And 3 things are important here. One is the matrix. Where are you going to sample? The water, the sediment, the biota, air, everything. That depends the method you are going to apply, right? And also this will depend the tools. Are you going to use a net, a bucket, maybe, or maybe just visually count the pollution? And also depends, for example, the size class. Are we focusing on microplastics? It means like large items like a plastic bottle, or we are going to measure the microplastics? All these questions are very important to decide the method that we are going to apply every time we go there and monitoring. So your data is comparable each time. When it comes to sampling strategies, we have different types. This is decision number 5. Once you decide where to sample, how to sample, the frequency, and everything, there are 3 means to prepare your survey. One is a random sampling. Every point has an equal chance of being chosen. That means we collect data from random places in an area. Another one is the systematic sampling. In this case, we have a fixed pattern. For example, we take one sample every 10 meters along a beach. And the other one is the stratified sampling. This is dividing, for example, the beach in 3 areas, one far from the water, maybe one more close to the water, and the other one more close to the water. And we sampled systematically in each area. So each strategy has strengths and weaknesses, but this depends on your question, what you are trying to find from what is your research question. In the case of the random sampling, this is unbiased, and it will give us very strong statistical power for the analysis of data, but it can miss maybe some rare data, like very low numbers. In the case of systematic, it's easy to do and gives a full coverage of the area, but it can miss very small scale patterns. This means like differences happen in a very short space. And in the case of stratified measurements, capture the zones at different, for example, distance from the water, but you need prior knowledge to understand how to establish the areas, the stratified sample. In this case, for example, the knowledge we can get is that we know, for example, from the tides, from the ocean, the plastic pollution might concentrate more close or more far from the water. And based on that, we can distinguish maybe 3 areas across the beach. And that is the information we need before planning the monitoring. So no strategy is best. The right one only depends on what you want to find. If you want to understand how the plastic pollution in a beach change with the tides, you will use a stratified sample. But if you just want to know how the concentration of the microplastics in the sand is presented across the beach, maybe you can use a random or a systematic. So now let's talk about something that many students forget. This is the reference sites. A measurement without a reference is hard to interpret. If you find, for example, 50 plastic items per square meter in Pantai Marina, this concentration of pollution is high or low? You cannot answer without a comparison. That means having a reference site. A reference site is a similar environment, maybe another beach, with low or no human impact. With a reference site, you cannot say this place is very polluted or not polluted. This is basically a comparison that will give us the information as a reference. This is something I will skip because maybe not much time. So now we'll give the activity number 2. I want you to spot the problem in the design monitoring. So we are going to read the scenario below very carefully. And then I want you to type one word in the chat, okay? The category that you can choose to detect what is the problem with this monitoring could be a time problem, a temporal problem, you put time in the chat, okay? If the problem is a spatial problem design, you will put place. If there is no comparison size, no reference size, you will type reference. And if you think the conclusion of the study is too broad from the data we actually obtain, you will put the problem is the conclusion. So the scenario says, a research team measured plastic on one Bali beach on one single Sunday afternoon in September. They count 4 items per square meter. And they conclude, Bali beaches has low plastic pollution all year around. So now let's try to think about this. They went to the measurement to take a measurement in Bali in a beach one single day in an afternoon, and they say we have this number of plastics per square meter, and they say the plastic pollution in Bali has very low pollution all year. What do you think could be the problem with this approach, with this monitoring approach? A temporal problem? A spatial problem? Maybe they didn't measure enough space, or maybe they don't measure enough times, or maybe they don't have a reference site, or maybe the conclusion is not good for the data they have. What do you think? Try to guess. You can type in the chat. Anyone brave? I will give you 1 minute to think, and maybe we can discuss together the data. Place. So far, everyone's saying place. A special problem? Yeah, the place. Okay. Anyone else? You can make mistakes. This is like playing. The idea is to discuss and understand the concept. Time, conclusion, good. Anyone else? Okay. Maybe well, we have some people saying spatial problem, a place problem, one for conclusion and another one for time, and 0 for reference. Okay. The interesting is that all of the real problem, all 4 are actually real problems in this study. But let me talk you about it. So there was no wrong answer here. In the case of time is a problem because they only measure one Sunday afternoon, 1 month, and that is only one tiny window. So plastic in the beach can change daily with the tides, with the storm. Maybe they have a cleanup activity. So a Sunday afternoon is particularly suspicious to say how much microplastic pollution we found, right? So many beaches are clean maybe after a Saturday or before Monday for tourism. And September also is the dry season, maybe very different from the rainy season. So we cannot say that the pollution is like the conclusion, is the conclusion is too broad. It says like pollution is all year. But with this time scale that we measure only one day, 1 month, we cannot say the pollution happens all year around. So we have a time problem. When it comes to the spatial, the place is a problem also because one beach cannot represent all Bali. Bali has a lot of beaches, right? Beaches which are near the river, beaches maybe in more touristic areas, or maybe other beaches are not so close to the city. So they will have different load or concentration of plastic pollution. In the case of the reference, also is a problem because they measure 4 items per square meter, but how do we know if this concentration is high or low? If we don't compare with a remote beach with no pollution, we cannot say nothing about this number, right? And when it comes to the conclusion, I think it's a dramatic problem, the worst one, because one day, one beach, only during one season, and the conclusion is Bali beaches year around, all year. This is a gigantic jump from one data to say all year. So this is very exaggerated conclusion, we'll say, for this data, right? So which one is the worst? Honestly, it's a matter of opinion, but here is my view. Conclusion maybe is the worst because the other three has flowed could be exist in different studies with an honest or modest conclusion. Maybe you only be able to measure a few times, but if you do conclusions according to that data, that will be acceptable. But if you only measure a few times and you make a grand conclusion and it's wrong, it's exaggerated, that's the worst thing that could happen in a research project. So this is a good lesson. We have that even a flow student, a student with problems, can be useful somehow. If the authors, the researchers are honest about what their data is actually showing, the real danger is when the conclusion goes too far beyond the evidence we have. So now let's talk about bias and uncertainties in monitoring. How we are doing with time? Okay, we still have time. Good. So now we talk about something every scientist must understand. This is bias and uncertainty. So here's the truth that most people don't like, and is that no measurement is perfect. Every measurement has three things. One is an error, random difference between repeated measurements. This means that one day I measure, the second day I do another measurement, or maybe one day I do many measurements. There will be an error between measurements, always, slightly, depending on the method, depending on the proficiency of the person measuring, there was always a slight difference. Also, the bias, systematic differences caused by the method itself. Sometimes the method cannot capture the reality in the best way. So that means that the method has some bias, and we need to understand that. And the third one is uncertainty. What you do not know about the true value of your data. That is the uncertainty. What do you know? What you do not know, sorry. So good scientists will not hide that they have error, bias, and uncertainty because this always happen. They would describe them so others can judge the conclusions that they found based on these limitations. So methodological choices acting as a filter of the reality. Think of your method as a filter, okay? It would only capture part of the reality in the environment. We have three examples with different contaminants. For example, we use a 5 mm mesh to capture microplastics floating in the water in a river, for example, but the microplastics will be missing because the pore size of the net is too big to capture microplastics. In this case, we obtain information from macroplastics, but not from microplastics. Another example could be a surface graph of lead will miss the deeper sediment layer. If we only take a measurement in the surface of the sediment, we cannot say about the information of the lead concentration in deeper sediments. Actually, in deeper sediments is where most of the lead will concentrate. So the method will define what fraction of reality we are measuring, what we can see, what information we can take. If you change the method, you will see a different result. And this is not a mistake. This is because of physics in the environment. So an example could be two studies, same river, but we find opposite results. For example, two studies in the Java River, a river from Java during the same year, but we found different results. One team, team A, used a surface DAO with a net, with a 500 micron mesh, very small, and they found particles around 2 particles per cubic meter of water. But another team were to the same place during the same year, but they used a water pump with a mesh of 50 microns, very small. So they found a concentration way much larger, 180 particles per cubic meter. In this case, this is like 90 times more concentration, but we are talking about the same river during the same time. Actually, both teams are correct, but they use different methods. So each method will filter the reality differently, in a different way. This is why comparing studies, when you are reading papers or research studies, you need to check to the methods, not just the numbers, not just the results. If you want to compare data, you need to compare what kind of method they use and how that affects the data they obtain. Another example could be if we want to measure microplastics, measuring at different depths in the water column. For example, microplastic in the water column, at the surface, we have most of the floating plastic concentration, but at deeper waters, maybe we will find fewer particles. And at more deep waters, we will only find a few particles sink because they have some organic matter attached to the particles during time and they get heavier, so they are sink. This is an example of how measuring at different depth will affect the number you will get from the environment. Another thing that we need to consider from the methods is the units. Units matter much more than people think. Particles, for example, per cubic meter of water volume, we are measuring water volume. There are only one convertible with method information. But the particles per square meter, per area, the conversion depends if we know the depth of the sample, because you multiply the depth for the surface and you will obtain cubic meters. So if we don't have additional information, sometimes we cannot compare results using different units. So when we read a paper or a publication or a study to compare with our results, we need also to check the units that they are being used. And if we have additional information to make the conversion to our units, if needed. And another thing is the detection limits. I don't know if you remember from previous lectures, we talked about the limit of detection. The limit of detection is the smallest smallest amount that your method can detect from one contaminant in a confident way. For example, if the limit of detection for your method is 1 microgram per liter of mercury, a sample in the environment, if you find that the data measurement says 0.3 micrograms per liter, this is reported as below the detection limits, but that doesn't mean zero. Below detection is not meaning zero. The mercury is there, still present, but it is below the detection limits of your method, so you cannot inform these results in a confident way. But that doesn't mean that the pollution is not there. So this is a common mistake that we can find in many papers, that they say there is no contamination, but when actually they just could not measure the level of contamination with the method they apply in the environment. So this is the 5 common sources of BIS monitoring. Let me show you 5 common examples. One is the selection bias. You only sample where it is convenient for you. Like always, for example, you measure near a roadside. This was a selection bias, because you will find high pollution because you are selecting actually a site that you know has high pollution. Another one is the temporal bias. You only measure maybe during one season, the one you are expecting more pollution, maybe the rainy season. Another example could be the observer bias. Different people count differently because they have different abilities. So maybe one person will find one number, and if another person goes and try to measure with the same method, they will find another number result, and that is called the observer bias. We also have to consider the instrument bias, because sometimes the equipment also have some differences in the values that they can give, depending on the brand, depending on the method that the equipment use. Maybe some people has an old equipment, another has a new model with different characteristics, different features, and the result will be different. That is something that we need also to consider. And finally is the reporting bias. Only the interesting results sometimes get published, and the boring results stay in the computer. But sometimes, many times happen when we talk about environmental pollution, that some studies, when they found 0 results, they don't publish that data, but actually that data is equally important as the ones saying that it's very polluted. So a good monitoring will be using the standardized protocols to try to reduce these 5 different types of bias. If you always use the same protocol in a standardized ways, that means that always the same protocol, the same methods, the same steps, you will avoid having this kind of bias. So how scientists reduce the bias and uncertainty? We have basically 5 standards practices. One is using calibrated instruments. That means that any instrument, for example, you want to measure the temperature, the pH of the water, the turbidity, or anything that you want to measure, you need to check these instruments regularly. This is called calibration. Every instrument has a calibration setting, and you need to check that regularly to make sure that the measurement from the instrument is correct. If not, you cannot make sure that the equipment might have a problem. So it's very important to check them regularly. Another thing is to apply standardized protocols, like I mentioned before. For example, in the case of plastics, we have many guidelines, international guidelines, like from GESAM, from UN, NOAA, which is from US, and so on. Maybe the BRIN in Indonesia also have their own guidelines. This is protocols already proved because they work. So if people want to start and study about plastic pollution, they go and check which is the proof protocols, the standardized protocols, and always tries to apply these kind of protocols. So your data will be comparable to other studies. If not, you go there and just do whatever you want. That doesn't make your data comparable. Or we need to also, another thing to consider is to train multiple observers to count the same way. Because if not, we will have the observer bias, remember? So if the people can train and trying to measure in the same way or almost the same way, you will reduce this bias this way, like training the people. And also an important thing is to take replicate samples. Sometimes it's not possible because of the cost or timing, but if possible, if we take many samples at the same site, it is better to reduce the instrument bias or the temporal bias or the selection bias by using replicates. And always try to document everything, even what things that you consider maybe not interesting. As much data as you can get every time you go on monitoring, it will give you an idea of what could be go wrong when you finally get the results. So the most honest sentence in a paper is, limitations of this study include blah, blah, blah. In that section, you will put all the limitations that your study has so people can understand why, how you get your data, and if it's comparable to their data or not. So one thing I want you to take clear about this is that bias is not a failure. Hiding the bias is a failure, is a problem. I want you to remember this one. Every monitoring study has limitations, but the good science, the good researchers will name these limitations. Bad science will pretend they don't exist, but a good scientist will inform. We get those results, but the limitations of these studies are these: 1, 2, 3, 4. That is the good way to inform science. When you read a paper, also always try to find out the limitations section, because sometimes it's often the most important paragraph in a study. This is not so common practice. Not many studies include the limitation sections, or maybe they don't have such a section, but they inform the limitations in the paper. That is a very good practice, and it's very good to understand and to make a full analysis of the information that you're getting from there. So now I will give you an example, a real example of our project in Thailand. A real monitoring project for microplastics. So we have one question and 3 matrices: water, zooplankton, and corals. And we did a coordinated file sampling campaign in Sichang Island. Sichang Island is a very popular island near Bangkok, maybe a few hours from Bangkok, in the Gulf of Thailand. So this is a coral reef conservation area, very close to a heavy populated coast because many tourism over there. So our team was asking a single question: how are microplastic distributed across the different compartments of this reef environment, this reef ecosystem? So one question, but we knew that one measurement will not answer the question. So we designed the 3 coordinated studies from the same project in the same field campaign. So each time we went there and measured surface water to measure the microplastics in the surface using a Houston net. Then we also took zooplankton samples to understand how the microplastics were in the water column and in the zooplankton in organisms. And we also, well, my colleague, she dives and she went there and took samples from corals in the bottom to measure the microplastic presence and accumulation in the corals. So in this case, we have 3 different matrices and 1 question. So let's look at the design. So we sampled this environment from August 2022 to April 2023. This cover in Thailand, the wet season, the intermediate period, and the dry monsoon. So, and both the spring and the neap tides, we also measure during the spring tide and the neap tide in the same place, during the wet season, intermediate season, and dry season. And we use measurements in 3 different matrices, so plankton, water, and corals. So we have 3 scientists working, 3 protocols, comparable protocols, but all coordinated at the same time. So for the surface water, we use a Newton net with a pore of 315 micromet mesh, standardized following the guidelines, international guidelines from GESAMP. Then for the zooplankton samples, we also use plankton nets to concentrate the plankton floating in the water. And then I took the copepods and I just eliminate the organic matter during some chemical treatments to find out if there was any microplastics inside of the zooplankton bodies. Also using standardized international protocols. So my data will be comparable to other studies. And in the case of the coral organisms, we have some chemical separation also of the organic matter. The coral compartments is more complex. We have like a mucus outside, then we have the tissue, and then the skeleton. So we have to develop a new method to understand how was the microplastic concentration in 3 different tissues. Why this is important? Because standardized methods and during multiple season sampling and during different tides comparison give us data that we can trust and we can compare with other studies and during time. So the findings, and here is what we found. Each matrix told a very different part of the story of microplastic in these coral ecosystems. In the surface water, we found microplastics abundances very different, very from one season to another. For example, the concentration was from 0.2 particles per cubic meter to up to 42. That's a big difference. So from very low numbers to more high numbers, depending on the monsoon season and also depending on the tide conditions. In the case of the zooplankton, this is the study I lead. We analyzed 750 individuals of one copepod, and we found constant microplastics inside all of the copepods. Very small particles. In this case, we found particles around the 50 microns. Compared to the ones we get in the surface using the method with using nets, we find particles from 300 micrometers or larger. The ones in the copepods are much smaller. And in the case of the corals, we could find microplastic in the mucus outside, in the tissue of the corals, and also in the skeleton, including in the calcium carbonate. We found microplastic inserted in the carbonate. So that's a very interesting information because we consider that maybe corals could be a long-term sink for microplastics. That means that the microplastic can, once they get to the coral skeleton, they cannot be released again to the environment and they will stay there forever. So what do we learn from this actual monitoring? That not single matrix will say the full story. If we only go there and measure the water, we will not know about what's going on in the zooplankton or in the corals. We will never find out, for example, that the corals can sink microplastics during a long term. If we only measure the zooplankton, we will find very small particles of plastic, maybe a few polymers, but it will not be a description of what we can find in the water, where we find very much more numbers of polymers, different type of particles, and even larger numbers. So, but together, all them, they reveal how the system behave, how maybe the microplastics behave in these systems. So this is some figures from each paper. In the left, you have the one of the corals and the one from the zooplanktons. You can see we use different type of corals, different species. In the case of zooplankton, we did the digestion. This means the removal of organic matters, and analyze the concentration of microplastics per individual. And in the case of the water surface samples, we found that the concentration was changing across time. And during the wet season, in this case, we find higher concentrations compared to the dry or intermediate season. And I think this time the hypothesis of the wet season was confirmed, because we were able to go there and measure more than one time. But in the case of the Vampacon River, if you remember, we could improve that because we only be able to measure one time. So now, why I'm showing all you this project and all these details and information? Because the logic of Sichang is the same logic you will use in your own studies that we want you to prepare as a final project. So 5 lessons that apply to any monitoring study is: you need to have one clear question that will determine what method are you going to apply. You need to measure, maybe, if possible, in multiple matrices, capture complementary parts of the system. Maybe you can measure in the water, maybe you can measure also in the sand. And also an important thing is to using standardized international protocols to make the results comparable with other studies. So you can say these values are high, they are not high, what is going on in other countries or in similar situations compared to here. And also a thing to consider is the multi-season sampling. If possible, sometimes it's much better to measure during different seasons too, so you can understand the change through time, the effect of time on the sampling and the decontamination. And also using coordinated teams, because sometimes you might have a small time, very low time, or a small budget. So working on teams will make you to have for one project a comprehensive data set that is more useful. So here is one of the bigger problems. Most of the studies worldwide cannot be compared. This is very common in the microplastic studies research topic. For example, in this case, in the Sichang project that I show you, it worked because we have 3 scientists that agreed to use similar protocols, comparable protocols, before going to the field. But this is very rare situation, not always happen. For example, across hundreds of studies of microplastics in Southeast Asia, including Indonesia, Thailand, Malaysia, many other countries, they are using very different methods. They sometimes using different mesh size for the Newton net, or maybe they report in different units. Some people report microplastic per cubic meter, other microplastic per square meter, or maybe they're measuring at different depths. Some people is measuring in the surface, some people is measuring underwater or in the bottom, or maybe using different processing techniques. All this change in the protocol will give a different results. So it's very important to use the standardized protocols I mentioned before, so you can make comparable your data with other studies. If data cannot be compared, that is meaning that you're wasting your time, because we need comparable data to make the policy-making process. So this is very important. For example, in this case, IMDOS, the initiative I mentioned before, is trying to solve this problem. IMDOS is the Integrated Marine Debris Observing System. It's a global initiative coordinated by different organizations, GOOS, GeoBlue Planet, UNEP also again, and GESAM from UN. The goal is by 2030 has a global interoperable monitoring system for marine debris. What does it mean? Means that people from different countries will be able to use similar methods so we can compare the data of plastic pollution, even if we are not measuring in the same place at the same time. That is very important. This is not easy task, but it's very important for the policy-making process because this is what it's all for, right? We are going there to monitoring, we invest time, we invest money, we put a lot of energy, but if the data is not comparable, it's not useful. So before we close, what time is it? Okay. Before we close, I want you to see the human side of monitoring, the constraints. One of them is budget. Sometimes we have a limitation of lab conditions, equipment, boats, money. Another limitation could be time. Maybe the research project is only for 1 year. Sometimes the project's during like 3 years, but depends. Environmental trends sometimes need decades to see what's actually going on. Another limitation for real researchers is the personnel, the people. Training a consistent team is very hard, it's very time consuming. You need to teach all your team to do exactly the same techniques in a good manner. So everything takes time. Also, the access to the study sites, maybe analyzing during the rainy season, the tides, you need permissions. You cannot go there because the weather conditions are not good during the rainy season, so there is no way to measure that. And finally, also the equipment, because not always we have the equipment we actually need. This is the ideal situation. You will have all the equipment that you need, but sometimes, most of the time, doesn't happen. So every published study, research that is done is the result of many different compromises. Understanding them is part of reading critically one paper, any research project. But considering all these problems, real problems, how researchers will handle these problems, this constraint? They will not stop the science, they will shape it. Here, for example, I will show you 5 strategies that real researchers can use. Some we use in our own work in Thailand. One example could be partner with local universities for equipment access. Sometimes one university has an equipment that you need, and sometimes they need another thing from you. So maybe you can do like a collaboration. That means that all universities will be able to do their work. Another thing is to combine the file monitoring with citizen science to extend the coverage. An example of citizen science is, for example, beach cleanup. When these NGOs making the beach clean events, what they are doing is also collecting all the information of the debris they collect from the beach, for example, and all that information could be very useful for researchers. Another thing would be use very simple methods. For example, using visual counts when the budget is tight. When you don't have much money to use very fancy or doing very experiments using equipments that cost a lot of money, sometimes you just need to use a reliable method that is easy to get, easy to apply, and you don't need much budget for that, and the information will be very useful anyway. Another thing that you can do to solve the problems that we mentioned before could be build long-term partnerships with communities for repeated access. That means that you make a partnership maybe from one collaborator so you can repeat the same measurement across times, or maybe doing this sharing the work, you know what I mean? And also combine the file data with satellite maybe data or drone observations. Sometimes is a good tool to use other technologies to get the data that you cannot get by yourself. So even if we have limited resources, that will not stop you to doing science. It will only shape the way you do it. But there's always solutions. So to end this lecture, let's do a final activity. We are going to use the reaction emojis from the chat. I know you like it. So I will show you 3 research questions, one at a time, and each question has 4 possible methods. Method A will be a thumb up. For answer B will be a heart. For answer C, you will raise your hand. And if you choose answer D, you will party. So 1 reaction per question. Okay? So for the first question, we have, are plastic increasing at Pantai Marina year after year? We have different options. The option A says that we use a method using monthly beach transects for 3 years using the same protocol. Another method was citizen science application doing daily counts of pollution for 1 year. The option C will be the same beach. They made a transect survey during 3 years, but each year a different team and a different protocol. And the option D is they decide to do 5 different beaches sampling once each in 3 different years. So different sites and different timing. So which one do you think is the best to see if the pollution, plastic pollution is increasing year after year in one site? Which one of the methods do you think is the most reliable one? A, B, C, or D? You can take maybe 1 minute to consider, and then you can start reacting with the emojis. Maybe you can put the emojis in the chat because I can see them. I'm not sure if I can see the reactions here because I'm sharing my screen. Good. We have one voting for option A. A beach transect for 3 years using the same protocol. That's a good one. Another one for A. Okay. Marco also says A. Raka say A. Anyone else? Someone for option B, C, or D? Okay, let's see the result actually was option A. So you did good. And here's why. The question is about the trends across different years. So you need many measurements with the same method over a long enough period of time to see the change. That's why we are going to choose a monthly beach transect for 3 years using the same protocol. That is the better way to understand if plastic pollution is increasing year after year. Very good. Let's see the question number 2. Which plastic types dominate in Banjir Canal Barat? We have 4 options. One is visual sorting of microplastics by color or in shape. The option B, the hard, FTIR analysis of plastic samples. FTIR is to detect the plastic polymer. Option number C is count items per square meter every month. And option number D is we do a manta troll with a mesh that capture all size of pollutants. So which one do you think will reflect which plastic type dominate? A visual sorting of microplastic by color of shape, a polymer analysis of the plastic samples, or shows counting how many items we have per square meter every month, or using a manta troll with a mesh. Option B, good. Anyone else? Option C, counting items per square meter. B. So we have 2 for B, 1 for C. Okay, most of you chose for the B, which is analyzing the polymer. That is very correct. But I saw some of you maybe choose option A, or maybe no one. I'm not sure. Maybe no one. The visual sorting by color and shape also give us a little bit of information, but the best option is the C, the B, sorry. So because we want to analyze the plastic pollution types, that means the polymers, polyethylene, polypropylene, nylon, polystyrene. The visual sorting by color and shape give us some information, but it's not the same information. So the option B is the best one. It will identify exactly what kind of plastic is dominating. And in the case of option C, it will tell you how many items are there, but you don't know what they are made for. And D is a good sampling method, but for other, without laboratory identification, you don't know what the type of plastic polymers you can find. So it's kind of incomplete. So maybe let's keep this one and try to finish before, because it's already time. So now let's see about 6 things to remember from today's lecture. I want you to take these ideas to home with you after today. One important thing is that monitoring and experiments answer different questions, remember. One is to see what is going on in the environment. The other one is to see what happens if I change this condition. Number 2, every study will start with a very clear question. You need to find that question, that it tells you what to measure, when to measure, how to measure, and where. Number 3, where, when, and how your sample changes what you find. All right, this is very connected with the number 2. And also, no measurement is perfect. We always will have some bias, and this is a really common problem in science. But as long as we are being honest about the limitations, the data will be easy to compare and easy to explain and interpret, not only by you, but for others, people reading your research work. And number 5, standardized methods is very important because it will let us compare our data across different sites, different years, and even different countries. So real world monitoring data is a compromise. These questions will shape the method. So now look at what you can actually do after this lecture. As of today, you can identify whether a study is monitoring or experimental study. You will be able to spot the design decisions, maybe what they want to understand or prevent. You can recognize any bias or uncertainty in the study if you pay attention to the methods and why they are important. You can also see why 2 studies of the same place might find very different results, and at the same time, they will be right. And you can also tell that a good monitoring from a poorly designed monitoring. You can now maybe have some tools to say this monitoring is well designed and this one maybe has some flaws. This is the foundation of science. Every environmental conservation builds on it, okay? So for the visitors joining us today and the people from Pollution lecture as well, you can visit and learn more about the GPML UNEP digital platform and also the Atlas of Ocean Microplastic initiative from the Ministry of Environment Japan using the websites in the screen. Also about the IMDOS initiative for harmonization of methods and the HESAMP guidelines for microplastic monitoring if you're interested in applying this to your studies or environments. And if you're interested in my MAPRA projects, also here it is, the website. I want you to understand that monitoring is one of the most accessible parts of environmental science and is at the same time one of the most needed. So for the students continuing this course, what is next? In lecture 7, we will talk about the research design. You will learn to put all this together to make a plan for your own study. This is for one of the final activities that you need to do for the environmental pollution lecture, okay? Maybe you will go to the field in Semarang, and maybe you will need to execute your own design, okay? This is something we need to discuss more in detail with Professor Dita, but probably you will need to make a monitoring design with everything we were learning from the last lectures. Also, try to reread your paddle article examples from the news and maybe make 3 questions you would like your study to answer in this kind of situations. Maybe some initial thoughts will be what would you measure, where, and when. And in lesson 7 is where everything will become a plan that you can actually execute here in Semarang. So thank you everyone for attending today. You know how now environmental data is made. Next time you can read for pollution research numbers about plastic or maybe other pollutants like mercury, bacteria, or anything, and you will be able to know what question to ask about how it was measured, okay? If you have many questions, comments, or any ideas, always you can send me an email to the one in the screen. I think we don't have much time now for doing questions, but if anyone has a question, maybe you can put it in the chat and we can shortly discuss about it. If not, you can keep your questions for the next lecture and I can answer your questions there too, okay? So if no one has any questions, I think we can conclude the lecture of today. Thank you very much for everyone joining. And just don't hesitate to contact me anytime to my email address, or maybe contact the Professor Tita and she will send me your questions as well. So thank you very much. And I hope you have a nice evening and end of day. Thank you. Love the
Thank you.
reactions.
Yeah, thank you, Dr.
Thank you very
Belen.
much. Lovely. Thank you, guys. Always enjoying to give these lectures. I hope you enjoy it as well. Okay, bye-bye. See you next time. Next Monday, we have another online lecture. Okay? I think at the set time, around this time.